All right. Okay.
Are you going to mute everybody?
I've got everybody. Sorry.
No, mute.
Mute.
Oh, yes, I will mute everybody. Yes.
Yeah.
Share. Okay, you should see the bacteria, green bacteria on a blue background. Yes? Good, okay. I can see 4 of you. So it's, I need to stop. So this is about the microorganisms, things that generally require microscopes of different sorts to be able to observe them. Now, I have to have a little bit of history to start with because it's such an amazing area. So the first viewings ever were 2 different people at about the same time. I don't know if they knew each other or of each other, but there was a guy called Robert Hooke. He was an Englishman, and he, oh, a spelling mistake, sorry, should have a U in molds. And he described the fruiting structures of molds, which I'll get to later, things like molds on bread and fruit. And this is going back to 1665. So even though you can see them, they're still considered as microorganisms. Leeuwenhoek, Antonio van Leeuwenhoek, a Dutch guy, he actually saw bacteria. He designed this initially, the microscope he designed, actually there are pictures of it around, a little thing, we put a drop of water in a hole, and he had a little pin in front of it, and he was able to see, he put whatever he wanted on that pin, and he could see through this drop of water, which later he put glass in the shape of a lens. And the first time he ever saw bacteria. And then from 1676 onwards, we started the study of microbiology. So they are microscopic or submicroscopic. Like microscopic, you need an electron microscope or a normal microscope, a light microscope, or even just a magnifying glass, which we take, all these things we take for granted these days. But in the 17th century, well, that blew their minds. Now, we have microorganisms that fit into this category of non-living. So we have prions, which are really deadly, and viruses, some of which are deadly. So they don't live. They have no living aspect to them. We have living ones, which are bacteria, we'll talk about fungi and molds with a U. And some of them are disease-causing, and we call them pathogens. And that's where the word pathology is used and whatnot. And but most bacteria, most fungi are fine. We use them, we eat them. They're part of our lives. Our skin's covered with them. We've got billions in our gut. Now, they are very, very important to our survival because they do modify as we change our diets and other things happen. So, and generally they are the ones that attack us, like the viruses, are very specific to hosts. Now, then I'll get into detail later. Now, these prions are disease-causing, and the word comes from infectious protein, and they are not bacterial or fungal or viral. There's no genetic material at all. They're just a protein. Now, we all have these prions. There are 2 versions. We have the good prions in our brain. And it wasn't until they knew about them quite a while ago, I don't know, 40, 50 years ago. But recently, the last 3 or 4 years, information is becoming out that they think there's an hypothesis that these prions in us has got to do with the laying of memory, our memory. So, and they're totally harmless. And so the problem is there's another form of this prion where the slight change in the structure, same amino acids, but the structure changes, the shape changes. And so it from being a normal prion, the word rogue is used. It really is nasty. So here's a diagram. This is what, I won't go into the biochemistry of it now, but these shapes, we know what they are. If you want the names, they're here. The amino acid, the alpha helix, and that's a beta helix. It's quite different. So this is the normal prion. We've got a large number in our brain. Everybody has. The diseased ones, which you hope you never get, is quite different in shape. This bit here is this bit up here, you can see, and it's changed its format, and then it causes problems. Okay, so what happens is when one of these nasties comes in contact with one of these, it causes the normal one to become a diseased one. Okay, and so even if you have just one getting into your brain, that's the end, because as they move around, they don't actually move themselves, but they get moved around, things happen. So what happens is they cause what are called degenerative brain diseases. And I'll just go through some examples and I'll explain a bit more. So scrapie is the name of the disease, this prion disease in sheep and goats. And in cows, it's called the mad cow disease, BSE. BSE is what is normally called, because bovine as in cow, spongy form, that is in the form of a sponge, and encephalopathy, the pathy means disease, the encephalo means brain. There's also one in humans. It's all the same, but it's called a different thing in different type of animal, the Creutzfeldt-Jakob disease. And then now, this is new, they think that there's a fatal familial insomnia, that people can't sleep and eventually kills them. Now, also in Kuru from the highlands of New Guinea is the same thing. It's called the shaking death. And I'll explain all this in a sec. And now they also think there may be some cases of Alzheimer's this is due to. All right, so what happened, and the names actually give it to you, if you think what you once I tell you, you'll realize when the good prions become bad prions or nasty prions, prions cause a diminution of the brain tissue. Then you end up with holes. So the brain in certain sections, and then it spreads out, will become holy, full of sponge-like. And that's why with the cows, it's called spongiform. Okay, and it looks like a sponge, the brain. And that happens in all. So first of all, with the mad cow disease, it came from the sheep, because farmers feed cows protein. And what they do, they usually have dead sheep or parts of sheep that they've gone through the abattoirs or whatever, they I don't know, they cook them, or whatever they do, they just mix them all up and into a powder of some sort, and then feed them to the cows, which the cows eat. However, if a sheep is sick, that they usually bury it. In England, some farmers didn't tell the abattoir or wherever that produces food, or the goats, by the way, also. They actually then fed the sick sheep to the cows. And that's how the cows got it. Because our digestive or any digestive system will not digest this particular protein. The protein's shape is such that it will not allow the enzymes to break it down, the proteases. They can't break it down, so it goes through, finds its way to the brain, and then makes the cow brain wholly, a spongy form. In humans, it's exactly the same, but we didn't eat, I mean, it will happen if you ate it. However, in the 50s or 60s, I can't remember, CSL was taking pituitary glands from dead people and mushed them up and made and cleaned it up, and they extracted growth hormone, which comes from the pituitary gland, and they injected that into people with height deficiency. They weren't growing properly, because these people's pituitaries weren't producing sufficient growth hormone, so they injected them. And some of them started getting CJD, Creutzfeldt-Jakob disease. And then they traced it back to the batch that somewhere along the way, one of the cadavers, the dead people, had prions. And they just use it as an Irish stew pot. You never empty it, you just keep adding to it. And that's what they did. So once they discovered that, the process changed. So and Creutzfeldt and Jakob were two doctors. I think one was English and one was American. They worked together, and this came through. The Kuru is interesting. In the 50s, when Australia took over management of New Guinea, they sent people up into the hills to see who was there. And they found lots of natives. There were all groups, but there were warring natives. And it turns out that they found these people, mainly the women and the kids, with Creutzfeldt-Jakob disease, which wasn't called then, but they were staggering everywhere. They couldn't do anything, and they were really, really sick. And it turns out that they attacked each, the various language groups attacked each other. And Kuru, by the way, is the language group. That's the name of the language, Kuru, that they found this first. And they were cannibals. So who gets the decent meat, the muscle? The men. And the kids and the women got whatever was left over, like the brains and so on. So they ate all that. And that's why the men were fine, they were healthy. A lot of the women were quite ill. So that's a little bit of history there. So viruses weren't discovered till 1898. Two guys, Loeffler and Frosch, Germans, that there was evidence of foot and mouth, that there was some infectious particle smaller than bacteria. Now, they knew about bacteria because that was discovered quite a bit earlier in the 1800s, but the foot and mouth was interesting, and they didn't know why, because I couldn't see it on the microscope. So they postulated this idea. Now, viruses can only be seen with electron microscopes, which weren't discovered from there maybe 30 years later, 30, 40 years later. Okay, so the Latin noun virus means toxin or poison, and that's where the names came from that time. So they're called submicroscopic, and then using an electron microscope. Now, what is important also is to get your heads around the size of what we're talking about. So viruses range at different shapes and different sizes, from 20 to 300 nanometers. So 1 nanometer is 1/1,000,000th of a millimeter. Now, if you can picture a millimeter in your head, and you can imagine maybe 100th of it or 1,000th of it, I think you'd be having problems. Trying to picture a millionth, I don't think any of us can really do that. And that's how small, so the smallest ones are around 20 nanometers. Okay, and what they do, they are specific. They each have host organisms, and they have evolved with the organism. We don't know what the origin of them is, but every animal, every plant has got its own viruses. So viruses are non-living. They can't reproduce on their own. They do not use energy of any sort. They don't metabolize anything. They do not grow. They are put together by cells, and that's the size they stay. But they depend on their hosts to reproduce. Okay, so they take over the DNA or the RNA to produce new viral particles. Now, they're not cells, and they're not living. We call them particles. And generally, the coating is protein, and sometimes there's a fatty layer as well, sometimes on the outside, sometimes on the inside. The genetic material could be DNA, or like our current inhabitant, the coronavirus, they are RNA, and that relates to the material, their genetic material inside this shell that they have. Now, on another talk I would give about genetics, I can explain in great detail the difference between them, but they are different. So this genetic material forces the host cell, remember they can't do it on their own, they take over a host cell, and the host cell then produces these viral particles, which then are allowed to escape. Well, not allowed to, but just escape and infect other cells nearby. So those with RNA, like a coronavirus, are called retroviruses. Okay, why? Because once they infect the cell, a normal virus would implant the DNA. It's a DNA virus, would go into the normal cell's DNA and then take over the protein production process. Whereas the retrovirus, like again, the cold virus is also a coronavirus. The retro means it does it in reverse. So what it does, it converts its RNA into DNA, and then it goes through the process of making new RNA. It's a complicated thing. How it evolved is amazing, but it's a very complex process, which it does beautifully in the cells. Okay, so I really don't want to go into the genetics because it just would take far too long. But you can always go back and look at these names like reverse transcriptase and retrovirus. And if you're interested, do a bit of your own research. Eventually, I'll get to do some more genetics, heavy-duty genetics with you. Now, the retrovirus, right? So I just here, it integrates its virus DNA. Oh, not again. Into the DNA of the cell. So I'll tell you this much, that in the nucleus of the cell, it's basically all DNA that's being done. But the RNA causes the cell out of the nucleus to produce DNA and then puts it into the nucleus. So then it goes, they reverse again. Complicated, but the end result is it replicates the retrovirus. Okay. By the way, the coat of the retrovirus is also DNA and some fat, but it's the nasty part is what's inside. So this is what some viruses look like. Sometimes like this. This one's the AIDS virus. All these things on the surface are what help it get into the cell or escape from the cell. So an AIDS virus is also an RNA virus, which is here. And it's got, you see here, reverse transcriptase. That's the enzyme that goes in and puts it together as a DNA, and then the host cell reconverts it. This is a very interesting one. This is a bacteriophage. The suffix phage here means virus. And this one, this shape, looks like a moonlander, is a virus that attacks bacteria, and especially things like E. coli, the bacillus, the ones that are rod-shaped. What it does when these feet land on the virus, it flips itself like that, and then the nucleic acid is pushed through into the bacterium. It becomes part of the system, and new ones are produced. The flu virus, coronavirus, has got two bits here. It's got the blue ones in the diagram are called neuraminidase, and the red ones are called hemagglutinin. And what it does, what these do, and exactly the same with the coronavirus, these things help it get into the cell and escape from the cell. I'm just showing you diagrams at the moment. Here's an amazing computer-generated diagram of the Ebola virus. Notice, not round, it's sort of snake-like, if you like, and it's got layers, and it's got everything in there. And this is 80 nanometers in diameter. It's 1400 nanometers long. And again, that's still very, very small. So what the coronavirus does, and I thought I'd spend a bit of time with what the coronavirus, because it's affecting us every day. It's these spikes, okay, and it's called corona because it looks like a crown. So the common cold, the flu, SARS, remember, severe acute respiratory system, was big a few years ago. MERS, 3 years ago, 5 years ago, Middle Eastern respiratory syndrome, and I looked that up. I didn't realize that the dromedary camels pass it on to the humans. The dromedary are the single hub camels, by the way. COVID-19, of course. Now, SARS-CoV-2 is the name of the virus, and we're using that. So this is what it looks like. Okay, so we've got the spikes. We've got the RNA in here. It's got a lipid bilayer, which is the same as in our cells, where the fat cells produce. So you get two fat molecules rather, producing a membrane, and then you've got a protein membrane on the outside, and then you've got, of course, the spikes. So, and here we have an electron micrograph of one of the influenza viruses, and it's about 100 nanometers, and both of these membrane and protein coat are visible here. So if you can have a look at the protein coat on the outside, and then there's the membrane, and inside there would be the RNA. This one here is the TMV, the tobacco mosaic virus, and these phages are about 900 nanometers, and they're only 9 thick, but they are these are the ones that attack tobacco leaves, and they destroy whole tobacco fields. And they basically move, I don't know if you've seen tobacco leaves, but they're very, very large, and they just rub against each other. And it's very easy to, apparently, to pass this virus on. So I'm just showing you these to show you the variety of shapes. And here's the MERS virus. Notice here, you can see the spikes coming out very clearly. This is blurred. It's an old one. I couldn't find a newer one. The newer electron microscopes will be much clearer than this, but I haven't been able to find a photo. Okay, so how do they replicate viruses? They take over. They literally take over the mechanism for the cell's protein and genetic production. When one of our cells reproduce, it's all processed, but the new nuclear material, the DNA is produced, with new proteins, and whole new cells are produced. So from one we get two. Here, the DNA or the RNA, whichever one we're talking about, it doesn't matter. It takes over that mechanism, so the cell will produce everything that the virus needs. And so I'm saying it induces the host cell to manufacture all the components, and then it gets put together by the cell, and you get these new fast particles which then escape. So this is a standard diagram. So it's used in year 12 books when we talk about diseases. So what happens, here's the bacteriophage. It binds, these feet bind to the membrane. Notice here, this neck has gone through, the RNA comes out. The RNA then is replicated by the cell, and it gets everything reproduced by the bacteria, if you like. It produces new ones of these, and they're in parts. Each part is produced separately. Then the cell, the bacterial cell, puts it all together, and then once it's ready to go, it's called lysis, where the breakdown of the cell, and the bacterial cell breaks down, and these bacteriophage are released, and the process starts again. Now, once they've been released, once they escape, from the cell's destroyed, and then they find a nearby cell. So here's another diagram where a cell with the spikes comes along, goes in, produces new ones, and off they go. I should mention that in mammals, I'm not sure about non-mammals, but in mammals, if one of our cells anywhere in the body is attacked by a virus, it sends out messages to nearby cells, just in the intercellular fluid, not in the blood necessarily, to say, I've been attacked by a virus, protect yourselves. And the cells nearby, which receive the signal, then change the structure of their membranes. So it'd be much more difficult for a virus to actually enter those cells. So we have, it's part of the immune system. It's called the second level. It's not specific to just a virus that is attacked. So here we have, again, another diagram, and there's a website here. So you can see it, all these viruses inside here, and then they're released. The flu viruses, again, as I said before, they're retroviruses, and they have a specific, flu's a bit different. And there are these two proteins I mentioned, the hemagglutinin and neuraminidase. What this one, hemagglutinin, requires for leaving the cell, but it leaves the cell, but it's still stuck to the cells outside. The neuraminidase is an enzyme which cuts this bit that's holding it back, so it allows it to escape. I think it needs the two. And there are 18 different subtypes. Now, I'm sure you've heard of 18 different hemagglutinin subtypes and 11 neuraminases. So the H1 to H18 is for the hemagglutinin, N1 to N11. And each virus is labeled as, I gave two examples here, H1, N1, H2, N3. So you will notice in with viruses that attack us, they often tell you what sort of flu it is, and that's what it refers to. I've really gone through this before, so I'll leave that. So when they escape, they need the hemagglutinin to be cut, okay? And it's cut by another, the neuraminidase. So what happens is, what happened back in the 90s, Monash and CSIRO and the National University constructed a drug called Relenza. I don't know if you remember it. It was a powder. And within 48 hours of getting the flu, if you knew that you got it, you breathe it into your lungs. It's like an asthma thing. The powder, you go in. And what it did, it blocked the neuraminidase. The cutting edge was blocked by this drug. So the cutting edge, the neuraminidase, could not then cut the hemagglutinin, and then the flu virus could not be released. Make sense? Okay. So once they knew the structure of the enzyme of the neuraminidase, they did the cutting, then they were able to produce a molecule that fitted into that enzyme, what's called the active site. And once that was done, then everything was good. And that by the way, is called rational drug design. Once they know what they need the drug for, and they know the shape of where it's going to go, then they can work out the shape of the medication or the medicine that they're going to use. In this case, Relenza. And rational drug design, it's probably about 15 or 20 years old, this idea, and Relenza has been on the market since 1999. So it's been a while. So what happens here? This is the activity here. So here's the cell and the virus that's leaving. The green things are holding, that's the hemagglutinin, holding it there. And then normally the neuraminidase, the word here is cleaves, cuts, cleaves it, and then off it goes. But once you've got what's called the neuraminidase inhibitor, which blocks it, in this case, these dark things, it can't cut, and there's no more viral replication because they can't go anywhere, and that works. Other viral diseases, the cold, the flu, chickenpox, and shingles. I hope you've all had your shingles injection post 70, if you are post 70. Very important, because if you did have chickenpox when you were a kid, the viruses hide in us, and something triggers them, and we get shingles, which is very, very painful and not very nice. Herpes is another one. AIDS, HPV, the papillomavirus, hepatitis, yellow fever, and viral meningococcal encephalitis. And what that does, it attacks the meninges. That's one of the layers of the brain on the outside. Let's go on to bacteria. Now, these are bigger. These are living. They think that the earliest life on land may have been the bacteria. No one knows. But they were there 3.22. This is, I took it straight from the article, 3.22 billion years ago, first life. Earth was formed around 4.5 billion years ago. So it took a while, 1.3 billion years, to settle down, to get some water, to get the land. Now, these single-cell microorganisms that developed very, very early, and they were free living, so they were independent, and they were also parasites. But the parasites probably didn't exist as such then, because there wasn't another type of cell, unless you get a small bacteria hitting on a larger bacteria. But the parasites refer to something a little bit later. They do not have a nucleus, and they are classified because of that into a kingdom. There are 5 kingdoms, and the first one is Monera, and they're called prokaryotes. Pro is the first of the group, and they don't have any nucleus. They don't have any membrane-bound organelles, nothing like that at all. So this is a standard diagram of multitude type of bacteria. All the different types of bacteria are in this one diagram. Inside this red thing is the chromosome. There's 1 chromosome. It's circular, but it's all scrunched up like that. There's a large number of these tiny round things called plasmids, which bacteria use to communicate with each other. There are ribosomes, these black things, and what the ribosomes do is where proteins are produced on them. Then on the outside, they have different layers. Some of them have a slime layer, some of them have a cell wall. They all have a cell wall, and they have a plasma membrane, which is made of fat. Okay, And then there's liquid inside called the cytosol. And some of them have a flagellum. Flagellum is whipping. They're like sperm tail. They whip. And they've got a number of strands of protein. And the proteins take it in turns in contracting. And that gives it the whipping process. They also have these pili, which are spikes which attach them to various cells. So they don't all have these. A lot of them don't have flagella or pili, or and their structures. You know, you can have bacteria with different structures on their membrane, and then you get different types of antibacterial antibiotics, depending on which bacteria they're trying to kill. Okay, so we've gone from the nanometers to the millimeters. So these you can actually see if your eyes are good enough. Okay, so they have no membrane-bound nucleus. They don't have any membrane-bound organelles. And everything that our cells do, it can do. Everything. And they exist as rods or spheres or spirals. And I'll show you some examples. And they're everywhere, even in New Zealand, where you've got the hot springs. They're there. They've got their DNA and their proteins that can withstand that heat. And they have a single strand of DNA, as I said, and these are called plasmids. Here's a similar diagram here. So, and that the cilia, I didn't mention before, and they just wave. I can tell you that we've got something similar in our trachea. Going from bringing material, phlegm, for example, mucus, from the lungs up into the throat and then into the stomach. So as we breathe, if you're, I don't know, somewhere with people smoking, you can see smoke because it's made of particulate matter. You breathe it in and it sticks to the mucus. This mucus is constantly, day and night, moving up. If you're going through a dusty area, you get dust stuck. And obviously you get microorganisms stuck in mucus on the way to the lungs. So it protects the lungs. And we have cilia. They're very similar to the pili. And these cilia, they're waves. They create waves inside, and everything moves up and into the esophagus and into the stomach, where everything's destroyed. Okay, so it's not unique to bacteria. They have a cell wall. Now, glycoprotein, glyco refers to carbohydrates, sugar, and protein. That's how they're made, and they're small. And I mentioned before, they generally attach to other bacteria, because bacteria can also reproduce sexually. That means they share some DNA. Here are some examples, and down the bottom you can see them, but the spheres, there are 4 shapes. There are spheres. You've heard of coccus, diplococcus, staphylococcus, streptococcus. The streptococcus are the ones that affect the chest and the throat. And if you remember, I don't know if they're still available, there were cough lollies called strepsils. Do they still exist? I don't know. And that's where the name came from. The streptococcus are the ones that they're into. The rods are called bacillus, the spirula, the spirulum, these things, and the vibrio. They're like commas. And I'm sure I'll put the diagram in the notes. You can have a look. So bacteria are reproduced by what's called binary fission. Now, fission is opposite to fusion. Fusion is when 2 things come together. Fission is when they separate. So what it does, it decides to multiply, to reproduce, which they do very often. They're just two new ones, and then they just separate. And this is a form of asexual reproduction. So it grows and it splits into two. So you have a bacterial cell, the DNA doubles over, and they separate, and then they move on. Now, and then they become independent of each other. Depends on the bacteria, because sometimes some bacteria stay in groups. And if we go back to here, hang on, this here, you can see these are in groups. So that's a streptococcus, that's a staphylococcus. They in the groups of them. These are more like grapes, bunches of grapes. E. coli, under ideal conditions of 37 degrees, like our bodies, where everything is there, you can do it in a beaker or somewhere, anything, keep it warm. Every 20 minutes, it will replicate. So it just grows and grows and grows. So you've got exponential growth under normal conditions. Now, some bacteria form spores, and these spores, like tetanus, they form another membrane under unsuitable conditions, and they'll just grow once they come out and grow once the environment changes, and but they can be like that for years and years. And that's one of the problems with tetanus, because it's usually found in horse dung, and when the horse dung dries, it gets blown around, gets caught on barbed wire or whatever. And if you scratch yourself on a farm, especially one with horses, the chances of picking up tetanus spores is very good. And that's why it's important to have our tetanus shots. Not that we go on farms, you can get other ways as well. So a spore, there's your basic bacterium, and there's a spore in there. Then there's a spore wall, and so peptidoglycan. The glycan is the sugar, and the peptido is the protein. So it's a protein-sugar combination molecule. It's got another form of peptidoglycan on there, and then you've got a keratin. Now, keratin, we are covered in keratin. Our fingernails, our hair is a protein called keratin. We have a layer on right through our whole skin, on the surface of the skin, of keratin, and it protects us. And it does the same for the spore, for the bacterium. And it could be like that for years. Now, we've got these facultative anaerobes, and they live in the absence of oxygen. But if there's oxygen, they can survive. So tetanus can survive, but it prefers the absence of oxygen. So if you get a scratch and you get a tetanus spore going under your skin, the skin is very poor in oxygen levels. And the tetanus bacterium comes out of its spore and starts to reproduce. Now, it does not move around. It stays where the scratch is. But what it does, it produces a molecule which we call a toxin, a poison. It's called tetanus toxin. And that moves around in the blood. And it causes problems. There's another sort called an obligate microbe, and it dies in the presence of oxygen. The staphylococci and the streptococci both cannot cope with oxygen. They get into inside the tissue and they stay there and reproduce. I won't say any more about that. But most bacteria are aerobes, and they do need oxygen to survive. How are they useful to us? Well, in food, lactobacillus, all these things here, yogurt, cheese, sauerkraut, all these things, even beer and wine, kimchi, sourdough, they all have lactobacillus. Lacto, by the way, refers to milk. Acidophilus, normal in yogurt. Both of them are in yogurt. It should be, if you eat yogurt, have a look at this side of the packaging. In the gut, we have our own microbiota, we call it the gut flora. We can have up to 1,000 species. I have a separate talk just on the microbiota. And we have huge numbers in there. In fact, there are 10 times, or is it 100 times? I think it's 10 times as many bacteria in us and on us as we have cells on our bodies, making up our bodies. There are also soil bacteria, which are absolutely essential for decomposition. Something dies, it gets decomposed by fungi and then bacteria, but the nitrogen fixation, where they take nitrogen out of the air and produce nitrites and nitrates, which are important for the soil. Now, we also use bacterial filtration in sewage. Now, sewage is the material that's in sewerage. So the stuff that goes through the sewerage is a sort of a system that the bacteria in there and the material is called sewage. And in Werribee Farm, if you know, on the way to Geelong, there's a huge place there. They used to grow the bacteria on grass there, and what they did, they opened these sluice gates, and they flooded these. They have quite a large number of paddocks. They used to. I don't think they do anymore. These paddocks with raw sewage, and the fluid sank, the liquid part. The bacteria are on the grass, broke down the sewage, and then everything just went through harmlessly into the bay. And they produced amazing nutrients for the grass, and then everything went through as the water, and some nutrients went into the bay. And Werribee Farm had lots of cattle, and because of the richness of the grass, they won all the cattle competitions at the Royal Melbourne Show every year. Some years ago, they stopped allowing the cattle to be shown at the show. It was unfair because of the system. I don't know if they still do that. I know the cattle did, but the actual thing. By the way, I don't know if you know that Werribee Farm is larger than Phillip Island in area. So we have about 10 trillion cells in our bodies, and we have about 100 trillion microorganisms in us and on us. So we have this relationship with these bacteria, and it's a symbiotic relationship. They benefit from us and we benefit from them a lot. So how do they help us? They digest our fiber. They produce vitamins like thiamine, which is vitamin B1, biotin, B7, vitamin K, very important for blood clotting. And they also, I'll come at that. They make up about 60% of our dry mass in the feces. And each of us has between 300 and up to 1,000 different species. Maybe the average around the world be about 500 different species. Some are good and some are bad. and depends on interactive and what you eat will affect the composition. It says here, I've got these figures, that about 99% of our gut bacteria are made up of 30 or 40 species. There are lots of others, but the bulk, the good ones, are there. And 99% are facultative aerobes. Okay, they're the ones that are happy to live without oxygen, but can live in oxygen. So that's important. How do they help physical health? Well, if you take antibiotics for too long, you start killing these guys off, and or something else destroys them, and you get dysbiosis. That means a badly affected biology in the gut, and it's associated. Oh, I think I'll turn my mobile off. It's my arm trying to get hold. Excuse me. Oh, there we are. Okay. Enough. So things like obesity, heart disease, stroke, abnormal cholesterol, type 2 diabetes, there seems to be a direct link with the dysbiosis in our gut. It also we know affects our mental health, and there's in Belgium they found that people with depression have constantly low levels of coprococcus. Now, copro means poo, shit, and coccus, and another type of bacteria here. And this Lister, by the way, was a very famous scientist called Lister. That's another story. Whether they took antidepressants or not, they were still depressed because of the lack of these two types of bacteria. And so what they've done not that long ago, it's now a common procedure of fecal transplant. So they ask for people to voluntarily provide good, healthy, fit people, provide their feces. The feces are tested for the type of bacteria that they have. They are then vitamized in a normal kitchen vitamizer, and they're put in packs, like as in the blood bank puts blood in packs, they put feces in packs, and then and then they transplant these feces into someone with this dysbiotic problem. And so you've got this natural microflora, as they're called, also, and they sort out the imbalance. Now, what sort of diseases do we have? We have gonorrhea with a bacteria called gonococcus. Now, the cocci are the round bacteria. Treponema pallidum, syphilis. These are very easily treated with penicillin. Clostridium welshi, the gangrene. Now, gangrene, this is a bacterium that likes to live without oxygen. Escherichia coli, that lives in our colon, and we get upper gut infections. We can get urinary tract infection. So when girls are little and they're taught to wipe their bottoms, they're taught to wipe the bottom away from the vaginas, because if they wipe towards the vaginas, they get constant UTIs, the urinary tract infections, with bacteria going into the vagina. It's not a nice thing. There are also respiratory illnesses like pneumonia and meningitis, and that can be caused by E. coli. What's going on here? What have I done? Oh, okay. And then we get streptococcus, I mentioned earlier, for throat infections, staphylococcus, like Staph aureus, golden staph, which are very common in hospitals. And Vibrio is cholera, that's one with the commas, and they have a little flagellum. And other examples like tuberculosis, typhus, plague, and diphtheria, typhoid, pertussis, that's whooping cough, dysentery, botulism. Botulism, by the way, is a bacteria that lives in canned food if the food wasn't heated to a high enough temperature. And what happens there is you usually know if a can that you have at home has botulism bacteria in it, is they're still living, and they don't need oxygen, and but they do produce a gas. And then if you have a can, the lid will be domed because it's been pushed up by the gas. If you have anything like that, do not open it ever. Send it into the health department. They'd love to know where the can came from and how many other cans have been produced with the botulism bacteria in it. Pneumonia is another one. Stomach ulcers have been found, or they're also called peptic ulcers. Well, they've had tissue lining broken down, so the acid in the stomach was affecting the actual stomach wall, which is mostly muscle. It's got 3 layers of muscle, so you can squeeze in different directions. But there's a very thick layer of I lost the word. Mucus, which protects the muscle, the wall. And there's a bacteria called Helicobacter pylori. Pylori refers to the stomach. It's got a valve called a pyloric valve. Now, they thought that the stomach ulcer was produced by stress, by anxiety. They used to think that. And what they recently found out that so even though we have a lot of people, about a third of people over 40 who are infected with this strain of bacteria, it used to be thought that it was purely anxiety that was doing it, and high stress people were getting peptic ulcers. But in 2005, 2 Australians, Marshall and Warren, were given a Nobel Prize for finding that it was this Helicobacter pylori that was doing it. And I think it was Marshall who swallowed some of this Helicobacter pylori, and he got the stomach ulcer, with a peptic ulcer. He treated himself with antibiotics, and it disappeared. So these days, stomach ulcers are treated with antibiotics because of the presence of this bacterium. And that was an amazing Australian discovery. MRSA stands for methicillin-resistant Staphylococcus aureus. And this is, again, it's very common in hospital, or it's also called multi-drug resistant Staphylococcus aureus. And they are resistant to some of the penicillins and vancomycin and cephalosporins. So people in hospital who are getting these who are sick and getting penicillins. Penicillins don't touch them. So, and that's why they call this methicillin resistant. Now, some people are not calling it methicillin resistant, but antibiotic resistant. That's what the M stands for. So it depends who you speak to, I guess. These bacteria are very difficult to treat. Okay, hospitals, nursing homes is where they're found because other people have got them. It's a closed environment. And now they've got a new one called community-acquired MRSA. It's contracted in community groups if people have them. So this is a white blood cell, a leukocyte, and this is a slightly colored electron microscope. No, it wouldn't be. It wouldn't be. No, it's a light microscope. It's a light microscope. They're big enough to be able to see. We also have these bacteria called mycobacteria. They're quite separate from all other bacteria because they look like molds and they behave a bit like molds. If you look at them on a petri dish, they just look like a mold, but they're not. They're bacteria. And by the way, myco is Greek for fungus. And these mycobacteria are responsible for things like tuberculosis. And that's called Mycobacterium tuberculosis. Leprosy. Now, the Buruli ulcer, which we have here in Victoria, it's also called the Bairnsdale ulcer. And the skin ulcer, and it destroys the skin. And they get these great big holes in the skin, which are very difficult to heal. Found in East Gippsland. Actually, the researchers, and it's actually been moving up the coast, Mornington Peninsula, Bellarine Peninsula. Up to now, they thought I added these two because now they've been found there too. So they're heading up towards us. Often people walk through bushland and they're wearing shorts, for example, and the legs get scratched. Some that often pick it, they can't pick it up. And possums are carriers. Mosquitoes bite the possums and pass it on to us. It's another way. And I put the date here, the 3rd of February this year. They just discovered this. This is brand new. It's a bit like the Black Plague, which the bacteria were carried by rats, but it was fleas that bit the rats, who then went and bit the humans, not the rats, the fleas, and that's how the people got the Black Plague. Okay. Now, bacteria pass on resistance to antibiotics to each other. And these, I mentioned plasmids right at the beginning, the little circular bits of DNA, and they're passed on. So here's a diagram of, here's a bacterium. This is its normal DNA. These are the plasmids, and they move around. And let's say this little reddish bit here is a section of DNA which codes for a protein which would allow them to be resistant to some disease, to some antibiotic. Okay. So even if one bacterium has, through reproduction and just mutation, is immune to a particular antibiotic, it will reproduce. And these plasmids are actually passed around between bacteria. They constantly communicate with each other. So it will pass on its immune genetics through the plasmid to another one. And it'll reproduce it and pass it on. And that's how they read the bacteria that have, even if there's one in your body, you'll be full of these guys, these bacteria, fairly quickly, because they reproduce every 20 minutes or so, or it varies on the bacterium, but fairly quickly. And then the doctors find that you're not getting better and you're getting sicker. Okay, so this is how they do it. It's, by the way, there's no brain in there. It's not something that they do consciously, just something they do normally. They share information. Now, when they grow, E. coli in particular, and they're bacillus, they're rod-shaped, under normal conditions, under good conditions, they grow they reproduce every 20 minutes. So it's an exponential growth. There we are, there's a lag phase initially. And once they get going, you get this exponential growth very quickly. This is in a test tube or in your gut. It depends how much space they've got. And then there's a stationary phase. They don't go anymore because they're getting to the limits of the use of their nutrients. And then they start to decline and they die off. This is in a situation where they can't get out, like in a test tube or a flask. And what happens is, as they keep growing, because they're living, they also produce toxins, which are their waste products. And it's their waste products here which kills them. Okay, they run out of nutrients and their waste, or their waste products. Okay, let's look at fungi. We'll move on now. The word fungus is mushroom in Latin. So there's a huge number of different types and different groups. There are the yeasts, the molds, and the mushrooms. So fungi are classified differently, separately from plants. Okay, so we had the bacteria, and they were prokaryotes. They do not have any internal membrane-bound organelles. The fungi are the next lot, but they are eukaryotes, which have like us, membrane-bound organelles inside the cell, and everything is very highly organized. So the fungi, they're not plants, they're not animals, they're not bacteria. And a major difference between them is that they have a cell wall made of chitin. Now, chitin is a protein, and it's pretty hard. It's different. We have keratin, and plants have cellulose. They have chitin. So it's a way of distinguishing. And the study of mycology is a study of fungi. So now, chitin is a form of glucosamine. Right? So glucose with nitrogen. But lots of other animals are have a form of glucosamine, and they make up the exoskeleton of crustaceans like lobsters and the exoskeleton of insects. Now, mollusks like snails have radulae, and these are scraping things, and they're very hard, and they scrape algae or things like frogs that they eat. And cephalopods are octopus. Now, the cephalobit means head. So their feet come out of their heads. things like the octopus, and their beaks very hard, made of this material, chitin. And it's been very useful in medicine because they can make sutures out of it, and they make now contact lenses and artificial blood vessels. It's tough. It's you can see through it if you do it a particular way. It's very useful. Now, I mentioned the eukaryotes as opposed to prokaryotes, which are the bacteria. So they have membrane-bound organelles. Sometimes I get ahead of myself. But they are more closely, fungi are more closely related to us than to plants. So that's why I put this in here. So here are the bacteria, all the different groups of bacteria. I'm not going to go through them. You have another form of bacteria here called the archaea, I'll mention later. And here are the eukaryotes, the plants, and then the fungi, and then the animals. So from left to right is the sequence of evolution. All right. So again, each of these goes into multiple layers. So you may not have heard of archaea, and you may not have heard of these other words. And this particular diagram I found was the best that I could find, but unfortunately, the equivalent of eukaryotes is prokaryotes. Pro is first, and that should be here. But my artistic skills on the computer are not that good that I could change that. Now, the structure of fungi. Most of them grow inside their food, and they produce these things called hyphae. And so they are cylindrical. They are 2 to 10 micrometers. Now, a micrometer is 1/10 of a nanometer. No. Sorry, the wrong way. A micrometer, sorry, is 1/1000th of a meter. Is 1/1000th of a meter. So we have millimeter, which is 1,000th of a meter, and a micrometer is 1,000th of a millimeter. Now, a human hair is because different people from around the world, different groups have different thicknesses of hair. So from very fine to very coarse hair, look, 18 to 80 micrometers, which sounds a lot. In reality, it's not very thick. And these guys are much finer than any human hair. And it grows from their tips. So here, there's a tip there. And what it does, it produces enzymes at the tip, which then digests the food or their food, whatever they're in. And let's say it's bread or a fruit or leather or whatever it is. And they digest those enzymes that produce digested, they absorb it and they grow. And so what they grow in is we call the substrate. So they digest it, they produce the enzyme, and off they go. Now, this mass, so one of these is called a hypha. A group of them are called hyphae. The whole lot is called a mycelium. And this mycelium spreads. And what you see, we see, of a fungus, we call the fruiting bodies. So mushrooms are the fruiting bodies of a fungus, but most of the fungus is in the ground. You don't see it. So here's a diagram. This is the mycelium here, and these are different types of mushrooms, but each of those are what are called the fruiting bodies, and they produce spores to reproduce with. and they're underneath there, under the caps. You know, you look at a mushroom, an open mushroom, you see black, they look like gills, and inside the gills, between the gills, are the spores. Sometimes if you have mushrooms at home and you bought them closed, and then after a while they open up, you'll also find tiny little black specks like fly droppings, and they're the spores. Now, you may have heard of fairy rings. Now, that in the middle, some time ago, there would have been one mushroom or one toadstool or whatever it was. And in the ground, it kept growing out. Every year, it grows and grows and grows. And particularly when the temperature's right, everything's right, it sends up the mushrooms or the toadstools, and then they die off. Now they've used up the nutrients, so they keep growing outwards. So this fairy ring is the next lot. Eventually that'll die off, and the next lot will be at the next layer. And the stories, of course, were that fairies were on mushrooms, and so on. So it's a very old name, mushroom rings. Here's a diagram of a bread fungus, a bread mold, and the fruiting bodies is what you see, the blue-green fuzz, and you know, you touch it and you get stuff coming out. They're the spores, the powder. Officially, that's called the sporangium up there. They're the spores, and the rhizoids are the mycelia. Okay. Very tempting, aren't they? So this is how they would grow, and you probably have picked up rotten fruit or rotten something, and just has collapsed because the mycelia inside the fruit has actually digested everything, and it just collapses. But it's important for the fungus to keep producing spores so it'll spread. Now, these are the, if you look at the spore on top of these, just a microscopic one, so they are the spores. And here's a different version. I just found something with gills. So the diagram of gills where the spores are for that. But these are for mold, these are mushrooms. So, so you get distribution at certain times of the year. With some of them, they wait for a windy season, and then they release them, and then they blow them away. And so every single spore can start a new fungus. It's incredible, the numbers here. And this is another form of asexual reproduction. Now, when hyphae of different individuals meet in the ground, or maybe even in an apple, an orange, whatever, they meet, they actually exchange genetic information, nuclear material. And that leads to a new individual with different DNA, or different DNA information. So they actually have sexual reproduction, so they get more variety. And you get some that are resistant to certain drugs, certain whatever, temperature, and resistant to poisons, because it's not the same ones reproducing all the time. They do exchange material and become different. So here's an example. Here the rhizoids, they're the which become the mycelium, and here you get the sporangium, and the spores are released, and that is just the asexual cycle. Okay, spores grow new ones. However, if you've got, you notice on the diagram, strain 1 and strain 2, they meet like that. They send out these little extensions, and then they form a new sporangium. It's called plasmogamy. I don't use that word, but it's a new zygosporangium, and they get bigger. Notice, those of you who've done genetics with me might remember when you're talking about DNA, about chromosomes, you have single DNAs, 1N. If you've got a full complement of 2, it's 2N. So the 2N here is from here. These spores, by the way, only have one single copy of each chromosome. These guys here have 2 copies, one from each of the different strains, and then they send up new sporangia, and each of those have single copies, but they've mixed them up. So we get a totally new type of mold, a new fungus. What sort of fungal diseases do we have? We have Candida, which is thrush. We have fungi that can attack eyes, nails, hair, especially the skin, and that's what they're called dermatophytic. Phyte means loving, as in keratinophilic. So these are the ones that attack keratin or the skin, and they also have ringworm. It's not a worm, it's just that they grow in a ring like the fairy rings. Athlete foot's another one, and they go under the skin, and that's where the skin keeps peeling off. There are agricultural diseases like rust, Phytophthora. It's also got root rot or dieback, and things like potatoes. And it was the Phytophthora attack on potatoes in the 1840s, which led to the Great Irish Famine. Millions of people died. It's also the time that a huge number of Irish escaped Ireland, went to America and Australia, and a lot of them ended up in South Australia. There's a very strong Irish community there. There's a Dutch elm disease is another one, where the elms, or a certain type of tree, they die in huge numbers because of that. Medicines, antibiotics like penicillin come from fungus. There are immunosuppressants like cyclosporin. Chinese medicine uses them a lot. We use yeasts a lot in fermentation, baking, and alcohol. We like mushrooms, most of us. Cheeses, if you like cheese, Stilton and Roquefort are straight fungi that grow through milk. Botrytis cinerea, noble rot. If you know anything about wine, sticky white wine, especially from Germany, the noble rot is the fungus that, the botrytis goes around each grape, and the mycelia are inside and converts the acid to sugar. So these highly, highly prized, particularly expensive wines, which I really like, are very sticky, they have a particular flavor from the fungus. Incredible. They're also used, of course, in decomposition, things that die. If we didn't have fungi, would there be dead organisms everywhere? But it's got, they recycle things. And we just break everything down. And pest control, we produce sprays now, which they eliminate or suppress things, various plant pathogens, things that attack plants like insects, mites, weeds, nematodes, the tiny worms, and other fungi. So they're sprayed with a particular fungus for them, and it kills them. And we also have symbiotic fungi that that rely. There are plants which rely on fungi around their roots to increase the water and nutrient absorption, and the fungi actually produce. The plant produces food for the fungus through photosynthesis. So they rely on each other. They're a very symbiotic situation. And it's very recently found, this is a new thing, that eucalypts, some species of eucalypt have fungi growing underground between them, and they communicate with each other through these mycelia underground. I don't know what they say, but apparently if one fungus starts getting eaten by a bunch of insects or animals, other trees nearby will start modifying their leaves somehow to make them not irresistible, make them I can't think of the word, deflect their being eaten. Okay, so there's messaging going through. There's another group, the archaea. They look a bit like bacteria, but they're not. But they're able to live in extremely hot situations and chemically aggressive environments too, and all over the world. And when they live in extreme things like hot springs and deep sea vents, they're called extremophiles. They like extreme places to live in. So I'm finishing off with a couple of cartoons. What's the time? Yeah, oh, good. It was a shortcut through the hospital kitchen that Albert was first approached by a member of the antibiotic resistance. Hey kid, want a superbug? Stick some of this in your genome. Even penicillin won't be able to harm you. And that, if you understand the idea of superbugs, the idea of antibiotic resistance, this cartoon is for you. I like this one. You're right. Oh, hang on. Scientists discovered a new superbug. It's wearing a red cape and blue tights. And ladies and gentlemen, thus ended the lesson. All right. And
thank you.
Thank you.
Thank you, Ruben.
Okay, so any questions, anything?
Yeah, Ruben, I would like to ask, for instance, I'm allergic to penicillin and flu injection.
Yes.
And I'm not quite sure what to do about it.
Speak to your doctor.
I did, and I don't know. I don't know either. So
that's the problem. But
yeah.
Now there are artificial antibiotics, as far as I understand, which can be used in the place of penicillin.
No.
Which, because people who are allergic to, my father, for example, used to, when he was a kid, food was a bit short, and if something was okay, it had a bit of fungal material on it, everybody still ate it. But when penicillin came through and was used, he discovered that he was very allergic to penicillin.
Right.
Okay, so my wife is allergic to penicillin. She wears a medical alert bracelet to say that. And so if she needs antibiotics, she gets other stuff.
But what about the flu injection? I can't have it. That's
I wonder what's in the liquid
that's right. I'm going to hospital, have it checked out, finally.
Good.
Yeah.
Good.
Okay, thank you.
All right. So, yeah.
Ruben, have you heard of a Q fever vaccine?
Q fever.
Yeah, my daughter, because she works at the zoo, and she was told she had to get a Q fever vaccine. And I was trying to look into it before I came to you, but I didn't have a chance to. But I
No.
thought I've never heard of that before.
No idea.
Yeah, no, and
Nothing to do with QAnon.
I'm not sure. She works with the animals. I mean, yeah.
Yeah, I understand what you say,
Yeah.
but I have no idea.
Yeah. Oh, I know what you're saying now. No.
Yeah. I don't know. The letter Q has now got other meanings.
Yeah. No, they're not into that kind of stuff. No.
Anna.
Maya, remember you've mentioned mycobacteria,
Yeah.
and you described it. It's derived from well, the carriers are the
Yes.
possums. Is that the same? It causes this is the same as they call flesh-eating disease.
Yes. I think so.
Yeah. Okay.
They are bacteria in structure, but the way they grow, they look like a mold.
Mm-hmm.
Yeah.
And Ruben, what about the house mold? Is it the same or different? Like
The mold that you. Oh well,
Yeah.
see if you get some moisture somewhere and you get mold growing, say in the bathroom, which doesn't have good ventilation, that's a mold like a mold. Yeah, just.
Well, it's hard to eliminate, though. It's very
You have to
hard.
eliminate them. Yeah. You can wipe them down with
Chlorine.
chlorine. Yeah.
Or lemon juice.
I don't
Lemon.
know. Waste of lemon.
Yeah, lemon juice. I heard that.
But it's
Is
a
it?
waste of lemon.
Yeah.
Waste of lemon.
I've got a lemon tree.
I think you'd go to a hardware store and get something.
Yeah.
Bleach works as well.
Bleach.
Bleach. Yeah, chlorine.
Sorry.
Bleach.
Yeah, well, that's chlorine. Yeah.
Vinegar as well is supposed to do that.
Sorry, Margaret.
Vinegar is supposed to kill the
Vinegar. It's acidic. Yes. It's a weak acid, but that does.
The problem is there is a source of moisture. We don't know where it comes from.
Ah.
So that's the problem. Yeah. So
Yeah.
we have to eliminate that. Yeah.
Some time ago, we moved into this place last year, but there was a big water problem, and we didn't know what the problem was, and we tried all things. We even put a new roof on, and it cost a fortune, but it had to be done, and it wasn't that either. It turned out that next door, we called in this guy, and he's called a forensic plumber.
No.
And he worked out that next door, which is about that much higher than we are, because it's a bit of a slope here, when they built the house, they didn't put any link to the storm water, which is, you can't see it, but behind me, in front of me, there's a fence and there's a lane, and the lane is the storm water drain. And ours just goes out, but theirs came into our garden and came up the walls. So we had to change our corner, the wood,
Water level, the water level.
No.
Skirting board.
wood. So Peter,
Skirting board. Is that what you mean?
skirting board. Must be getting tired. Okay, now our skirting, when it was swelling, they took it off, and it was moldy. I mean, so that's a normal mold as well, and it destroys everything and it holds it. Yeah, so we had a few problems. Yeah, but he's a wonderful guy, and in fact, he had his daughter with him, who's his apprentice. She's got a psychology degree, wasn't happy, so she did a teaching qualification. She taught for a while, she wasn't happy, so she wanted to be out in the air. So now she was in her last year of apprenticeship with him, and did a great job digging.
So can you pass on the number to me? Have you got the?
Of course, I can do that.
That'll be great. Thanks.
Yeah, his name's Andrew. Yeah.
Okay, thanks. I'll
Okay.
leave you.
By the way, I get no commission.
Oh, well, that's what it's about.
All
Thank
right.
you.
Okay,
Thank
so
you.
on that note, I will leave you. And unless something else. Oh, sorry, Judy.
Judy.
Unmute yourself, Judy. Judy, unmute. Press the space bar down.
Yeah. Are your notes online?
Sorry?
Are your notes that we were
Oh,
looking at?
after today, yeah, in the next couple of days, I'm recording.
No.
Yeah, I'm still recording. And once I'll download it and then I'll put it online. Yes,
Yeah, 'cause it was a lot of information to absorb.
of course there is. And
No,
it's mostly in the notes anyway.
The notes were sent with the email.
Yes, yes.
The email that I sent on Sunday has
Yes,
got
I
the
got
notes
that.
attached.
It was
Yes.
attached to the
Okay.
email.
Yeah.
Okay,
So-
I'll have a
So double
look.
click on those. But
Thanks, Ruben. See you next time.
Thank you.
Thank you very much.
Thank you,
lovely to
Ruben.
see you again.
See you next time. Thank you, Ruben.
All the very best.
Don't
Thank
forget to stop
Lovely.
recording.
What?
Stop recording.
I will when we stop. Yes. Bye-bye.
Bye.
Bye.
Cheers, Ruben. Bye, everybody.